Hari tu kita sampai first line defense dah. So kita akan tengok untuk yang second line defense. Okay. Basically yang untuk phagocytic cell ni kita dah cover. Tapi tak apalah, kita just go through it again. Okay, so internal defense ni is known as second line defense. Okay. So it is triggered when the body has been, the pathogen has entered the body. Okay. So the pathogen managed to penetrate the first line defense lah. So there are four internal defenses, which is the first one is phagocytic cells. So these are white blood cells that has the ability to engulf pathogen. Okay, so you have several different types of phagocytic cells. You have neutrophils, for example, eosinophils, macrophage, okay, and also dendritic cells. Okay. And then the second internal defense is antimicrobial proteins. Okay, so actually kita akan tengok nanti. So basically, antimicrobial protein ni it consists of proteins that will be that the protein will be activated, and then it trigger a mechanism that form pores at the membrane of the pathogen and causes the formation of pores and causes water and also ion to enter into the pathogen and causes the pathogen to burst and lyse and destroy lah. Okay, and then that is one example. Okay, and then the third one is natural killer cells or NK. So the NK cells will destroy infected body cells. Body cells that are infected by pathogen. And then the last one is inflammatory response. So the mechanism we are going to look at later lah. Okay, so the first one, phagocytic cells. So this type of defense is known as the cellular counterattack. Okay, because the phagocytic cells or phagocytes detects the pathogen and attacks them. Okay, so the phagocytic cells will circulate in your body fluid. So once it encounters the pathogen, okay, so the phagocytes will engulf the pathogen because it is carried out by phagocytic cells. So the process is phagocytosis, engulfing the pathogen. Okay. So this is the mechanism. So phagocytic cells or white blood phagocytic cells, phagocytes, okay, white blood cells that has the ability to engulf, they will recognize the pathogen. Due to the pathogen on its cell surface, they have cell markers that is made up of polysaccharides, and then the phagocyte engulf the bacteria, and the bacteria is surrounded by vacuoles lah. So and then the vacuoles fuses with lysosome in the cells of the phagocytes, and then the lysosome produces nitric oxide and other gases to poison the pathogens, and then the lysozyme within the lysosome will degrade the bacterial cell wall. Okay, so this picture, it shows you the mechanism of how phagocytosis occurs. Okay, so you have here a cell extension of the phagocytic cells. It forms pseudopodia, which is cell extension that surrounds the microbes, right? And then so microbes are engulfed into cell by endocytosis or phagocytosis lah. And then it forms vacuole, all right? And then the vacuole fuses with lysosome, and then lysosome containing nitric oxide, lysozyme will digest the microbe into debris, and the debris is excreted through exocytosis. Okay. So these are the mechanisms. So vacuole and lysosome forms and fuse, and then toxic compounds and lysosomal enzyme destroys microbes. Okay. So these are the four types of phagocytic cells. You have the neutrophils. So as you know, neutrophil is the most abundant white blood cells. And then you have eosinophils. It has limited roles in phagocytosis because the main function of eosinophil is to destroy multicellular parasites. Okay, and then these two cells, macrophage and dendritic cells, once these two white blood cells engulf the pathogen, it will trigger the activation of the third line defense. Okay, so this is a second line defense. But if these two cells, macrophage and dendritic cells, engulf the pathogen, it triggers the activation of the third line defense. Okay. So macrophage. So macrophage or macrophage, okay, so it is known as, they are known as big eaters. So they are large phagocytic cells, and then they develop from monocytes. It means that the monocyte will mature to become macrophage. Okay, so they will circulate in your body fluid, your blood, your lymph, interstitial fluid. You can also find macrophage in the alveolus, for example. Okay, they reside permanently in organs and tissues. So you can find macrophage in the spleen, in the lymph nodes and other tissues of the lymphatic system, to fight off the pathogen. So engulf bacteria, viruses, and cellular debris, including those in the lungs. Okay. All right, and then the second one is neutrophil. So it is the most abundant. So when you are infected by virus or pathogen, so the one that will come first is the neutrophil. They are the most abundant. They will circulate in the blood. So signals from infected tissues attract neutrophils. So the neutrophil engulf and destroy the microbes. The next one is eosinophil. So can be found in mucosal surfaces lining the digestive, respiratory, and urinary tracts. They have low phagocytic activity. And then, so this is the function of the eosinophil. Part, it can engulf the pathogen. It can destroy multicellular invaders, multicellular pathogens such as parasitic worms and blood fluke. So how? So the eosinophil will position itself against the parasite body and destroys the parasite by secreting destructive enzymes, okay, that damage the invaders. So dia boleh buat dua lah, okay, destroys multicellular pathogen and also carry out phagocytic activity. Then the next one is dendritic cells, okay. So they roam in tissues such as the skin that are in contact with the environment, and then they will migrate to the lymph node after they have encountered the interact with immune cells to stimulate adaptive immunity. Yang tadi saya cakap, kalau dendritic cells, they engulf pathogen, dia akan defense ataupun panggil dia juga adaptive immunity ataupun acquired immunity. Alright, so dendritic cells perform macrophage function in most tissue, which is to ingest the microbes, which is to engulf. And then once the dendritic cells engulf the pathogen, so it becomes APC, antigen presenting cells. Okay, so what is the function of APC is that it will activate helper T cells, and the T cell will activate cytotoxic T cells. So the cytotoxic T cells, basically it involves in the cell-mediated immune response of the third line defense. Okay, cell-mediated immune response is to destroy infected body cells that are infected with specific pathogen. Okay, and then the so cukup lambat, right? Okay, so ini adalah antara soalan yang selalu keluar lah. So you have to give examples of phagocytic cells. Okay, ada 4. Okay, so the next, the second component of the second line defense is you have the antimicrobial proteins. Okay, so pathogen recognition in mammals triggers the production and release of antimicrobial proteins, which is peptides and proteins that will attack or kill or inhibit the growth and reproduction of microbes or disrupt membrane integrity of pathogen. So dia ada 2 cara. Okay, satu dia akan uh, inhibit your body cells from being infected by pathogen. Ada mechanism dia, kita akan tengok sekejap lagi. And then lagi satu adalah it forms pores, okay, at the surface of the pathogen cell membrane and causes the membrane of the pathogen, the cells to lyse, okay. So examples of antimicrobial protein, you have interferons and complement system. Interferons adalah untuk chemical yang terlibat dalam poin yang pertama ni. Okay, so this involves the interferon, and the complement system terlibat dalam ni, disrupt membrane integrity. So the function of the complement system is to form pores at the plasma membrane of the pathogen. Okay. So interferons are proteins that inhibit viral reproduction. Okay, so they are secreted by virus-infected body cells. So basically, your body cells that are infected by the virus or the pathogen will produce interferons. Okay, and the function of these interferons is that it will find healthy body cells and prevents that healthy body cells from being infected by that pathogen. Okay, so stimulate uninfected cells to produce antiviral proteins that inhibit viral reproduction. So maksudnya kat sini, sel-sel yang sihat akan hasilkan antiviral protein. Okay, so what is the function of antiviral protein? It is to limit cell-to-cell spread of viruses. Okay. Other types of interferons activate macrophage and this enhances their phagocytosis. Okay, so it enhances the phagocytosis carried out by macrophage. Okay, so basically you have this diagram. So the first diagram here, it shows you cells that are infected by virus. So you have here the virus, the pathogen. So the pathogen will enter into the cells. Okay, and then selalunya kalau virus ni, virus is different from the genomic composition of human, for example. So human we have DNA. Tapi kalau virus ni dia ada RNA, single strand nucleic acids. Okay. So this single strand nucleic acids, the RNA of the virus will activate, okay, gene within the DNA of the human and causes the gene to be activated and undergo transcription and translation. So transcription produces mRNA and then translated into the interferon protein. Okay, so the interferon protein will be secreted from the infected body cells. Usually the infected body cell will be destroyed. Okay, and then this interferon will find healthy cells, and then the healthy cells will be activated. The gene will be activated and then undergo transcription and translation. Transcription produces mRNA, the mRNA produces antiviral protein. Okay, antiviral protein. So the function of antiviral protein is to block viral reproduction. So ada 2 molecule kat sini, interferon by infected body cells, antiviral proteins by healthy cells. Okay, so it prevents the healthy cell from being infected by the virus. Okay, so the next one is the complement system. Okay, so this is still under antimicrobial protein. Okay, so the second type is complement system. So it consists of about 30 proteins in the blood plasma, function to fight the infections. So they will circulate in in inactive state. So mula dia inactive, okay. So how to activate? So they are activated by molecules at the surface of the microbes or pathogen. Okay, so bila dia encounter the pathogen, dia akan diaktifkan. It will be activated. Okay, so the activation of the complement system results in a series of biochemical reactions. Okay, so once it is the complement system is activated, they will coat the surface of the microbes so that it is easier for the macrophage to engulf, therefore enhance phagocytosis of the pathogen by the macrophage. So basically, bila complement system ni, protein ni dia dah salut surface microbe tu, dia akan menjadikan microbe tu inactive, tak boleh bergerak. So bila dia tak boleh bergerak, inactive, so senanglah macrophage datang and then engulf. Okay, so another mechanism adalah, so they will insert themselves into plasma membrane of the pathogen and make little holes. Okay, lubang-lubang pada surface plasma membrane of pathogen, and once pores has been formed at the plasma membrane of the pathogen, water and then ions can enter into the pathogen cells, into the cells of the pathogen, and causes the pathogen to burst and then lyse, destroyed lah. Okay, so dia ada dua cara lah posed by the complement system ni. Coat and then make little holes. Nanti kalau kita tengok dekat acquired immunity untuk humoral immune response, humoral immune response yang akan menghasilkan antibody. Okay, so antibody pada third line defense tu nanti dia juga akan aktifkan complement system. Tapi ini yang sekarang kita tengok adalah untuk innate immunity. So maksudnya ini untuk general. Kalau badan awak diserang oleh ini pathogen, ini adalah antara mekanisme yang akan terhasil, akan berlaku. Okay. So complement system also function in the inflammatory response. Okay, so as you can see from the diagram here, so the brown structure here is the plasma membrane of the pathogen. So the blue structure here are the complement system. So it enters, okay, into the membrane of the pathogen and causes formation of the little holes, okay, and then you have fluids and ions can enter into the microbes and then causes the microbe to swell and then burst. So the next one is the natural killer cells, so or the NK cells. So NK cells will destroy tumor cells and virus infected body cells. It induces apoptosis. What is apoptosis? It is programmed cell death. It causes infected body cells to be destroyed. Okay, so the NK cells patrol the body, attach to the diseased cells, form a hole in the cells, release two types of chemical, which is interferons and also granzymes, and lead to cells death or lysis. Okay, so this inhibit further spreads of virus or cancer. The NK cells are produced in the bone marrow. So NK ni persamaan dia adalah sama dengan cytotoxic T cell pada third line, tapi NK pada second line. Okay, second line defense, which is yang tadilah second line defense. Okay, so ini adalah bawah second line, tapi dia mempunyai persamaan dengan cytotoxic T cell pada third line. Okay, so mechanism, okay, so bila once the NK cells releases perforin, what is the function of perforin? Is to it is inserted into the membranes of the target cells and it forms pores. Okay, and then after pores has been formed, the NK cell release the second chemical, which is granzyme. So the granzyme enters the pores and activate proteins in the target cells that induces apoptosis. Kalau awak baca buku Campbell, salah satu lagi mekanisme macam mana granzyme ni masuk dalam sel adalah melalui endocytosis. So dia ada dua cara, melalui pores produced by perforin, satu lagi adalah melalui endocytosis. Okay, so you have here the picture. So you have the target cells. So these are the body cells. This is the body cells that is infected by the pathogen. So you have the NK cells. So the NK cell will secrete perforin, okay, macam lebih kurang sama mechanism dengan complement system tadi, okay. And then after pores has been formed, granzyme will be secreted and enters into the infected body program cell death. So as you can see here, you have macrophage that will come and then digest the cell debris of the infected cells, and also once the infected body cell has been destroyed, it exposes the pathogen, okay, in the infected body cell, and also the pathogen will be engulfed by the macrophage. And then the next one is the last one that falls under the second line defense. You have the inflammatory response, okay. So it is triggered by physical injury or infection by microbes of tissues, infection of tissues lah, okay. So kalau macam kena tercocok dengan pin ke, so the pin is contaminated, so obviously it brings in bacteria, for example, into your body, okay, so it triggers the inflammatory response. Salah satu lagi contoh inflammatory response adalah demam, fever. So fever is an example of systemic inflammatory response. Dia berlaku pada keseluruhan badan awak. Seluruh badan akan jadi panas, okay, inflammatory. So that's why nama dia adalah inflammatory due to the heat produced, warm, okay. So it is characterized by redness, warm, swelling or edema, and the skin. It brings in infection by pathogenic bacteria. And then you have a type of cells which is, we call it as mast cells here. So mast cells at infected or injured site releases histamine that have two functions. So the function of histamine is that it causes vasodilation of local blood vessels. So the blood vessels at the infected area will vasodilate. Okay, vasodilate to bring in more blood to the infected area. And then it also increases the permeability of nearby capillaries. Okay, so mast cells here, you have to remember that it contains vesicles. Inside the vesicles, it has histamine. Okay, so histamine will trigger this to occur, vasodilation, and also increases permeability of blood capillaries. So histamine, I add here, it is inside mast cells, okay. So it is important inflammatory signaling molecule stored in the mast cells. So what is mast cell? It is connective tissue cells that contain granules for secretion. Granules are specialized vesicles that contains the histamine, okay. So you have vasodilation that occurs due to the secretion of histamine by the mast cells. So the vasodilation of local blood vessels increases blood flow to the injury site. So this causes the area to become red. Why red? Because you have more blood flow to that infected area, okay. So red, and then warmth, okay, because more blood will carry more metabolic heat to the tissue, okay. So heat. So that's why the mechanism is called as inflammatory response. Dia akan menyebabkan keadaan yang sekitar kena infected area tu jadi warm lah, okay. Inflammatory, right? Red and warm. Okay, so next, capillaries that has increased permeability and engorged with blood leads to the release of fluid, blood clotting elements, and phagocytes such as monocytes and neutrophil to the injured area. So bila berlaku vasodilation, lebih banyak darah flow to that area, so lebih banyak tekanan yang berlaku dekat blood vessels tu. Bila lebih banyak tekanan berlaku kat area infected tu, akan berlaku apa? More fluid, okay, to flow out of the blood vessels and causes the tissue to swell up, okay, tissue swelling. or edema. So when the tissue swells, it causes pain. Okay, so pain because of the pressure on nerve cells ending on the region due to edema. Okay. Okay, so you also have blood clotting elements. So the function of blood clotting elements, it will wall off inflamed area to prevent or delay pathogens or toxic from spreading into the surrounding tissue. So maksudnya bila kat tempat tu kena infected, akan ada juga blood clotting element. So dia akan prevent microbes tu daripada merebak lagi ke tempat lain dalam badan awak. Okay. And then neutrophil. So tadi kita tahu kalau if you are injured, if you are sick, the most abundant white blood cells that will be present is the neutrophil. Okay, so neutrophil will migrate from the blood to the extracellular fluid or interstitial fluid that bathe the surrounding tissues to carry out phagocytosis. So the neutrophil secrete another signaling molecules that will attract monocytes which will differentiate into macrophage to carry out phagocytosis. Okay, so these two cells, neutrophil and macrophage, dua cells ni dia akan keluar daripada blood vessels, dia akan pergi kepada dalam interstitial fluid and dia akan release cytokines, okay, signaling molecules, alright. So the function of the cytokine is to increase more blood flow to the injured area and causes that will lead to redness and also warmth, okay. So ada dua lah tadi. Yang first akan menyebabkan vasodilation semua tu adalah histamin, okay, daripada mast cell. Yang lagi satu signaling molecule adalah cytokine daripada neutrophil dan juga macrophage. Okay. So phagocytosis by the phagocytic cell leads to the formation of pus. That is a mixture of dead pathogen, tissue cells and phagocytic cells. So what is pus? It is a fluid rich in white blood cells, that pathogen and cell debris from damaged tissues. Dia macam tenanah lah. Okay. So here is the diagram to show you how it occurs. So you have pin. Okay, obviously the pin is not sterile. It has microbes. It has pathogen. Okay. So at tissue injury, mast cell releases histamine. So this is the mast cell. So mast cell will contain granules containing the histamine. So mast cell releases histamine to the surrounding interstitial fluid and causes what? Vasodilation. So vasodilation causes blood vessels to dilate and bring more blood to the injured area. Okay, so it causes the area to be red. Okay, and then it brings more metabolic heat. It causes what? Warmth. Okay. So macrophage release signaling molecule cytokines that increase local blood flow. Another function of mast cell tadi apa? Dia akan increase the permeability of the blood vessels so that more white blood cells such as neutrophil and macrophage can exit the blood vessels and enter into the interstitial fluid. Okay. So blood capillaries dilate, become more permeable, allowing fluid containing antimicrobial peptides to enter into tissues. Okay. Ini yang tadi lah dalam second line defense yang nombor 3 tadi. Okay. Signaling molecules attract additional phagocytes. So the white blood cells here, you have the macrophage, you have the neutrophil, will engulf the pathogen that tries to enter the body, and then you also have what tadi blood clotting elements, okay, that prevents the pathogen from spreading to other parts of the body. So engulf the pathogen and then debris from dead white blood cells leading to the formation of pus. Okay. And then next, phagocytic cells, macrophage and neutrophil ingest pathogen, cell debris, and the tissue heals. Okay, so when you are injured, obviously you will feel pain. So apa benefit dia? What is the benefit? The pain will cause you to move less. Okay, so that it will promote tissue repair. Okay. So yang ni yang tadilah saya cakap pasal demam tu. So fever is an example of systemic inflammatory response. Okay. Systemic inflammatory response. Systemic, it means that it occurs throughout the body. Okay. Seluruh badan awak tu panas. Okay. So toxins released by pathogen and virogen produced by activated macrophage leads to an increased body temperature. So selalunya kalau demam ni suhu dia adalah lebih pada 37 point, kalau dalam termometer saya 37.5 lah. Okay. Tapi kat sini 37.2. Tapi kalau macam suhu dah start lebih pada 36.8, so akan dah rasa nak panas-panas badan dah. Okay. So 37.5 selalunya memang dah demam lah. Okay. So the benefit of fever, one is promote phagocytosis by phagocytic cells. Okay. Second, inhibit the growth of some pathogens by stimulating the liver and spleen to store iron so that blood iron level is reduced. Okay. And then the third one is accelerate tissue repair by speeding up chemical reaction. So suhu panas tu akan menyebabkan satu, promote phagocytosis, dua, accelerate tissue repair, yang ketiga ni dia akan menyebabkan liver awak store iron lebih. So maksudnya iron dalam awak punya darah akan berkurangan. Kenapa dia pilih iron? Sebab iron ni it is a nutrient that is essential for both microbial pathogen and the mammalian host. Maksudnya kat sini iron ni dia bertindak sebagai, dulu kan kita dah belajar pasal enzim cofactor kan. So iron ni dia bertindak sebagai cofactor pada enzim yang terlibat dalam metabolic activity of cells. Okay, tak kisahlah dia sel pathogen ke, dia sel host cell ke. So bila iron ni kurang, sumber iron ni kurang, so the bacteria cannot get enough nutrients, okay, so it cannot carry out proper metabolic function, okay, because less iron is present, okay. Okay, so kita pause dulu kat sini kejap. Safa, ada soalan tak? Okay, saya ajar seorang tadi. Yep. Nampak? Dah. Ni ironnya. Okay, bila badan awak panas, okay, fever tu dia akan menyebabkan awak punya, masa awak demam tu, liver dengan spleen, dua organnya akan berfungsi untuk simpan iron. Okay, iron ni diperlukan sebagai nutrien dalam tindak balas yang normal dalam sel-sel, tak kisahlah ada sel macrophage ke, pathogen ke, ataupun sel dalam badan awak sendiri ke. Okay. So kenapa iron? Sebab iron ni dia bertindak sebagai cofactor. Ingat tak chapter enzim? Enzim perlukan cofactor kan, supaya enzim tu boleh berfungsi dengan baik. So bila iron tu kurang dalam awak punya badan, maksudnya kat sini pathogen tu akan ada kekurangan sumber untuk dapatkan iron. So maksudnya kat sini pathogen tu, sel pathogen tu tak boleh lah nak buat tindak balas biological process dengan semula. So maksudnya pathogen tu tak boleh nak survive lah sebab tak ada iron dalam badan awak. Bukan tak ada iron, kurang iron. Okay.